In this video, we are going to talk all about infrared spectroscopy and infrared radiation and how we can use this to deduce a structure of a chemical compound. So to begin with, we need to remind ourselves what infrared radiation is. So it is found in the electromagnetic spectrum just outside of the visible range. So it's found right here in our electromagnetic spectrum. Now, within this region of the electromagnetic spectrum, certain bonds will absorb this kind of radiation. And when it absorbs this kind of radiation, it causes them to stretch or bend. So this stretching or bending, we can actually visualize and see what that is. And that is going to give us some information about the final structure of our chemical compound. So just to give you a little bit of an idea of what infrared spectroscopy is then, you have this machine. So the picture shown here is the typical setup of an infrared spectrometer. And basically what you do is you put your sample in this little container here. It comes in you put it in this really long tube. And then with the computer, what you do is you set it up to pass through the entire electromagnetic range of infrared radiation through your sample. And as that goes through your sample, you then collect the information, that absorption of the energy, and it gets then put out as a printout. And so we're going to take a look at what some of these printouts look like from this information. So it's important to keep in mind the printout of the spectrum is going to show which frequencies, and when we talk about frequencies, we actually talk in terms of units called wave numbers, which is related to the frequency, are being absorbed. And so it's a really kind of neat spectroscopy technique that we can use to determine a lot of information about a chemical compound. So I mentioned earlier in this video that when we shine light in the infrared region on a chemical compound, it's going to cause certain bonds to stretch and bend. So when you're thinking about bonds in this sort of context, you want to consider a bond to be like a spring. So you can pull and stretch and bend those different bonds. And what I'll do is I'm going to post this video in the description on YouTube. I'll post a link to it as well as in our course material. And you can just take a look at it and see the kinds of stretching and bending that happens for different types of bonds. You don't actually need to be able to name what those different stretches and bends are. It's just kind of neat to see the different ways that you can visualize what this looks like. Okay, so we're talking about all of these different vibrations. So one thing I haven't mentioned so far about infrared spectroscopy is that the only bonds that are going to interact with infrared radiation are polar bonds. And that's a really important point to keep in mind, is that we're only getting information about those polar bonds. And the intensity of the absorption is going to be dependent on the polarity. So the more polar the bond is, the more absorption we are going to get. So we get a little bit of absorption from some of the non-polar stuff, or depending on how it's connected to polar bonds. But then those polar bonds are going to be absorbing really, really big. So this diagram here just gives you a little bit of an indication of the types of wave numbers. And wave numbers are in centimeters to the -1. And some of the different types of bonds and stretches and stuff that you see in an infrared spectrum. So, for example, Down at the high end here, you get things like OH and NH bonds that will absorb. Whereas a little bit further down in the middle here, you get things like C double bond O bonds absorbing. So there's information and there are tables of data that we can match up the types of bonds to specific absorptions within a spectrum. So here's one such data table. This is given in the IB data booklet. So you do not need to memorize these numbers, but you do need to be able to interpret what they mean. So if you kind of just take a look, the first sort of 4 here are all carbon halogen bonds. And so they're occurring at wave numbers down between 490 and 1400, depending on the type of halogen that is attached. Then you've got things like CO bonds that are part of alcohols, esters, and ethers that are happening in the 1050 to 1410 range, and so on. So take a look through all of these. The ones we use the most are this CO bond. We use this C double bond O, which is for aldehydes, ketones, carboxylic acids, and esters. We also use the OH bonds. And if you notice, there's actually 2 values for an OH bond, and that's because it depends what else it's connected to. So if you have an OH in a carboxylic acid, it's happening around 2500 to 3000 wave number. And then the last column of this table also gives you some information about the intensity of the band. So you're going to see really strong, really broad peaks showing up in a spectrum. Whereas this other OH bond that's in alcohols and phenols happens a little bit further up. So it's around 3200 to 3600. It's also strong and broad, but it's not going to be as broad as the OH in a carboxylic acid. So we're taking the position as well as the shape of these peaks, and we're interpreting them and matching them up with pieces of information from our structures. So it might sound kind of complicated, but we're just going to go through and run through a couple of examples to show you what it looks like in terms of a structure and how to match things up. So this is a very typical infrared spectrum. This one is for propanone, which has a methyl group on either side of a C double bond O. On this axis here, you have the percentage of transmittance, which is really just a measure of how much is being absorbed or how much is being transmitted through your sample. So anything that comes down and has almost 0 transmittance means that it is absorbing almost all of that infrared radiation that is going through that sample. So we call that a peak, or say that it is a very, very strong band. So you see lots of bands in these as well, and you don't have to go through and interpret every single one. What's important is that you go through, and if you're given the structure information, it's really just about explaining what band corresponds to what part of that structure. So for example here, the one I circled, this really strong band that occurs at around 1700 wave number corresponds to our C double bond O band, right? It's in that range of 1700 to 1750, and it is of strong intensity. And it's a ketone. Okay, really that's the only band that gives us a good amount of information here to interpret the structure. I think the other thing to point out is that if it wasn't a ketone, if it was like a carboxylic acid or an ester or something, well, maybe not an ester, but you would see other bands, and the region you would want to look at is here especially, because that's going to give you information about whether it's a carboxylic acid or not. Okay, so this one's a really easy example. Let's go and interpret another one now. So this one is for ethanol. So we got a CH3, CH2, OH. And what you should notice right away is this really big broad band here, which corresponds to our OH vibrations. And it's more in this range of the 3200 to 3600. Okay, so that is likely from an OH that is part of an alcohol and not part of a carboxylic acid. And the way that you could double check this if you didn't actually know what that structure is you could look for the really strong band at around 1700, because you would expect to see that if it was a carboxylic acid. You kind of have a small peak here, but it's not big enough to be a carboxylic acid. So it's pretty safe to say then that that is an alcohol and not a carboxylic acid. All right, our last example here is butanoic acid. So this is an example of a carboxylic acid. We have that C double bond O and the OH. And you can see this really broad, big peak here around the 3000 range. And you also get this really strong peak here around the 1700 range. And so both of those correspond to and fit the data for a carboxylic acid. You got this one here, and you've got the carboxylic acid one here. So really, again, it's just matching up pieces of that infrared spectrum in order to be able to determine what the structure is. Like I said before, you do not need to interpret every single peak. You're going to find the ones that fit your data and that explain your structure the best.